Hello, everyone. I am going to take us in a little bit different direction than what we've been hearing. So I am an epidemiologist at Ancestry, so I didn't even think about my disclosures, but my disclosures are that I work at Ancestry. I'm on the personalized genomics team, and we have an Ancestry DNA science team. There's about 30 of us at this point. So today, I really want to focus on kind of what Dr. Ben mentioned in our work in distant genetic ancestry and some of our new work in this more novel, recent, fine-scale population structure we've been observing and really try to tie that to some of the implications for precision medicine. So briefly, before I get into that, I just want to provide you guys with a little bit of information about the resources we have at Ancestry. So the company was founded in 1996 with a really simple mission to preserve records to help people understand more about themselves and a little bit more about where they came from. So I really like using this picture. This is a picture of an indexing project in Italy. And when we went to get the records, we found that they were being stored in an unused urinal. And so what we do is take these records, we restore them, scan them, index them in a way where they can contribute to family, through your family history research. So currently, we have over 2.7 million subscribers. We have 90 million family trees, 20 billion records in these trees, in billions of profiles, and we have over 330 million photos and stories that we're working with. And then in 2012, Ancestry launched Ancestry DNA, and currently we have over 6 million individuals genotyped. So what I want to talk about today, I'm sure I'm going to go over concepts that are very familiar to all of us, but I just want to talk about germline variation in the context of the way we discuss it at Ancestry, so we're all on the same page. And then get into a little bit more of our work in the distant genetic ancestry, and then the ethnicity estimates that we provide, as well as this more novel work in the recent fine-scale ancestry detection, and then hopefully tie that into the relevance to precision medicine in providing a framework for you guys to really think about this population structure in your work. So we collect DNA through saliva. We use array-based technology to look at over 700,000 points in your genome. And we are looking at these single nucleotide polymorphisms, or these SNPs. We're really just looking at the nucleotide differences between individuals at a single DNA point. And as we know, DNA isn't inherited by these at each nucleotide, but more in chunks. And so a lot of our research also focuses on haplotype work. And in the context of this talk, we can just think about haplotypes as a large group of these bases that are inherited together. So here I have noted the AG would be the two alleles in this SNP, and then I've noted them in a large haplotype block. And you can share haplotypes with individuals because of common ancestors, and you can also share haplotypes with other individuals because those haplotypes are common in a specific ethnicity. So the genetic pattern of these SNPs and of these haplotypes among populations are really driven by human history and by migration patterns. So through migration, I'm not going to get into a lot of details here on population genetics, but through historical migration, there are events that have occurred that have made some haplotype structures more similar and others very different. We really utilize the differences in these haplotype structures to identify populations that are differentiated. So, I'll be referring to that as population structure throughout. So looking at this population structure at different time scales using the SNPs and haplotypes afford us multiple opportunities. The large shared haplotypes or lots of shared regions in the genome we can utilize to identify recent shared relatives. So what you're looking at are, maybe it's close family. So your cousins, who you're related to in Ancestry's large database. Conversely, we can use the smaller haplotypes or some of the SNP frequency differences to identify a larger global population structure where we're thinking hundreds to thousands of years ago. And a lot of what our work is focusing on now is somewhere in between. So it's trying to leverage the medium shared haplotype to identify populations on a closer time scale. So what we're looking at here is within the last few hundred years to see what we can really start to tease out of our data. So I'm going to start initially by focusing on the global population structure. So how we estimate and identify ancestral structure in ancestors living hundreds to thousands of years ago. So I wanted to show you first, I guess I'll leave the pointer, an ethnicity estimate that we are providing. So this is mine. I am 31% Irish, Great British, you're from Europe West. And so what I want to share here, actually we'll grab this, is we have these polygons that represent like the larger regions that we're very confident in, and then we have these lower, the ones that aren't colored in are the smaller portions of DNA, with our more low confidence regions. So how we work at getting these ethnicity estimates is by utilizing a reference panel that we have worked to build. So our reference panel has genotypes from over 26 global populations. And you can see in this visualization, these are all of the different regions where these populations are. And what we do is compare your DNA to the DNA of this reference panel to really understand the population structure and be able to provide that estimate of your ancestry proportion across these regions. It's very important to us how we build these reference panels out. It's important to think about how you build out a reference panel to represent populations that existed thousands of years ago. So in what this image is here is a picture of a family from Mongolia who has kept their records on a stone. They are individuals, we search out individuals with deep documented roots in different geographic regions of the world. And this family was involved in some work in our Sorensen Molecular Genealogy Foundation, work with them, and this family has kept track of their family history forever. And this is how we're able to really build out the specifics of this reference panel. So as Dr. Bem mentioned, a lot of our focus does work in identifying like to what level we can start to identify these groups. So initially, in our first version, we had West Africa. But after work and after really trying to build out this reference panel and understand any bits of information that could provide genetically distinguishable groups, we were able to build that out to 6 different regions in West Africa. So these are 6 genetically distinguishable groups within the West African region. So I wanted to touch briefly on how we do this. This is a figure from principal components analysis. And I won't get into too much detail on that, but if you think about it as, well, first, initially the colors represent countries. They have, like, for example, here, this is Mali, this is Nigeria. And then you can think about each point as an individual. So this analysis really teases out independence. So what you can think about are the dots that are closer together, or the individuals closer together, have less distinct genetic ancestry versus the ones farther away, where you can start to tease out more distinct genetics. And then what we really like to point out here is that a lot of this work mirrors geography. So we are able to really start to capture more of this structure across the more detailed in West Africa. So then we were able to take this and turn this into regions that we can then provide in these ethnicity estimates to let our individuals know a little bit more about themselves and take a little bit more ownership in their ethnicity estimate and understand more about where they came from. And then this information, I think a lot of you may be aware of this level of structure. This global population structure is something I'm thinking probably a lot of you think about in your work. some may not, but what I really want to point out here is that there's a lot of previous research that addresses that this continental level population structure can impact association analyses. So in this study detecting association case controls while addressing for this population stratification can lead to associations detected that have nothing to do with disease if that information is not taken into consideration. So you can really start to capture false positives in these analyses if you aren't thinking about this global population structure. As I pointed out in, say, West Africa, for example, those are allele frequency differences that allow us to identify those groups, which can also cause false positives in the association work. So in this study, Accounting for Ancestry, something to highlight here is not only can you get false positives, but if you don't take the structure into account, you can also wash out any effects that you may be able to see. So you're really decreasing your power in your genetic study, and for a more robust study, it's important to adjust for this population structure. So in this last paper, just really pointing out that this is a primary consideration in the study of genetic determinants of human disease. So as I mentioned, this is known in the GWAS, in context of the GWAS world and some of the work being done, but in the context of precision medicine, it's definitely something to think about if it's not already currently involved in your work. So that is more of the global population structure. But now what I'd like to focus on is some of the more novel work that we're doing on our team in identifying these populations on a closer timescale. So we say between 5 and 20 generations, we're thinking more, within the last few hundred years, what can we start to identify with over 6 million individuals? What can we really start to tease out with the smaller shared haplotypes? So we hope here to bridge the gap between your ancestry estimate or your global population structure and your recent ancestors. How can we really start to bridge that gap? So one thing I want to say about DNA matching overall. So this DNA matching here is referring kind of to that larger haplotype. We do go through our database, and we are able to tell you how you are related to the other 6 million people in the database. So the chance that a random pair of individuals share enough DNA to be considered fourth cousins seems to be a relatively small number, 0.0001875. But I would like to put that in a little bit of context here. So say we have 200 people in a room. The number of ways that I could go through a room and pick 2 different people out of 200 is 19,900. So what would you guys, any guesses here? The chance that there's at least one pair of genetic fourth cousins in a room of 200 people? It's 97.6%, so it's very high. So we really work to utilize this information to see what we can start to understand about population structure. So this is what I was explaining. What this is taking, for the matching, your genetic data, matching it on the entire database, and estimate this relationship. But what we decided to do is move forward and take the entire database on the entire database. So I'm going to talk a little bit about how we do that. So if you think about this giant hairball of people, we have, you think about the circles as nodes or in the individuals, and the lines are the relationships identified in between these individuals. And I do want to point out these are all individuals who have consented to research. This is a giant hairball of relationship. What I will point out about this one in particular is this is only with 75 individuals. And so if you can imagine scrolling this up to millions and millions. But what we do here at this point is run a community detection algorithm to really let the data tell us what the structure is within. And we're able to tease out clusters of individuals that are more strongly connected and related to each other than the rest of the groups. So this individual here is solely connecting through his DNA with the orange group, but we also have individuals who are connecting between 2 different groups. And so I will point out at this point, this information has been done solely using genetic data. So we've taken our data, we've built a network, and we've found these communities. I'll refer to them as genetic communities, that's what we call them. And we've done this with shared haplotypes, using the DNA matching to identify the populations. But now we're sitting there with these clusters across our data thinking, okay, who are they? How are they clustering genetically? Who are in each of these groups? So what we're able to do at that point is really pull in all of that other information that I was describing at the beginning, and start to use pedigree information, start to use information about people's ancestries, their ancestral birth locations, and what their estimated ethnicity is, to start to tease out detailed stories of these populations. So we can go through these clusters and we annotate them with pedigree information. This culminated in a paper this year, actually, in Nature Communication, where we were really able to identify pretty amazing aspects of US demographic history. These groups consist of multiple communities of African ancestry, communities in points of immigration where you have German, Scots, Irish, all in one genetic community, but due to the time scale that we're able to look at this, they're clustering to be more similarly clustering in the United States with each other. And so you can see, we work with historians to really start to build out these stories and understand what migrations were occurring at what point, what geographic events that were causing isolation. But you can see there's also a fine, you can see the Mason-Dixon line, I think, which is overlaid in the publication. But it's pretty interesting in the last couple hundred years to see what we're able to tease out. So future work, since that publication, we've been able to identify over 300 of these genetic communities all around the world. So in what I wanted to say is now, well, one step before I get to it, but every day new people are coming and taking ancestry kits and taking their DNA tests. And as our database grows, we really need to work out a way to put or estimate where these people may fall in these genetic communities. And so we have a process where we build classifiers, and we're able to use estimated genetic relationships to assign these individuals to a genetic community. So we have, in these over 300 or so identified communities, I wanted to focus on a few here. After some conversations with Dr. Bim, I went through the communities and I really wanted to find, these are examples of some of our Hispanic, Latino, admixed communities. So these are communities that, after we've looked at the breakdown, are enriched for Hispanic, Latino ethnicity. So you can see we have, you can go through and look at where these communities on the website. You can start to play with where the communities are, and kind of read some of the stories of the migration patterns. But here's North America. We have Mexico and northern Mexico. These are all sub-communities within. So each time you click into this, you find more fine-scale regions, deeper and deeper into the genetic communities. One thing I wanted to point out about these, though, so I pulled one in specific. This is Northwest Mexico and Southern California coast. What the dots are that you're looking at are dots that represent ancestral birth location enrichment. So we've taken from the trees the ancestry birth location that people have provided, and we run an enrichment analysis comparing it to all other outside of that community. So the ancestral birth locations have fallen nicely into where within these genetic communities, and the lines, well, first see the bottom here, there's a time period that you can click through. I picked 1825 because this was a major migration period for this community, but these lines represent migrations that we've been able to identify in the pedigrees. So you can see a lot of movement from this community up through California, especially here in this region, which looks as it could potentially be Sacramento. But what I wanted to point out is here's another community in about a little later of a time scale that is genetically distinguishable from the last. So the important thing that I want to point out here is these groups overlap geographically. The migration patterns are similar. They're also in California. So as Dr. Ben was saying, these groups are both checking non-Hispanic white or Hispanic white or whatever on their self-reported ethnicity, but they're genetically distinguishable. So in this fine scale structure, I really do want to reiterate it is something important to think about in the context of precision medicine. And the same goes for the African American communities. These are Louisiana Creoles and African Americans. You can dive into more detail. This is an example here. I just wanted to put it in, and what you would see is working with historians who specialize in, and we've reached out to multiple historians because each historian specializes on a certain group, and so we make sure that we can get the most accurate story that we possibly can on these migration patterns. But this is kind of what it would look like. You get the overview of the population, and you can click through the time period, and you can watch the migratory patterns that we are able to observe in our data. For certain people, if you did have a family tree, you can see where your ancestors fall within this pathway also from your tree. But I did want to point out these two groups, two communities geographically overlapping that are genetically distinguishable, along with Mississippi African Americans, the same here. This also breaks down into smaller communities. And an example of a migration pattern in the North Carolina African Americans. So this was in the 1925s. You can start to see northern migration patterns from some of the southern African American communities. So what are the implications for this work in precision medicine? This is a very complicated table. Don't pay attention to all of the details. I am going to bring your eyes to what I'd like you to focus on, but this is supplemental from that Nature Communications paper. But what we did was take SNPs from the GWAS catalog, and if you're familiar with this, it's a way where everyone puts their most significant findings from their GWAS to make the findings publicly available. So we've taken SNPs from that, and we've been able to look at these diseases. This is just an example of a few of the diseases that we've looked at across some of these genetic clusters. And what you find is, for example, for type 2 diabetes in the New Mexican, Northeast, and Western Mexico community, the frequency of this variant is 18.8%, while looking at outside of that community, the frequency is 5.2%. So you are noticing some large frequency differences in these groups. For the African Americans, I circled prostate cancer here to look at, and high density lipoprotein. So again, you see 41% for this variant within the community versus 9.5% outside. And something very striking to me is for this prostate cancer variant, it's 55 times more common in this African American community than it is in the rest of our database. So some more recent work that exists that's identifying some of the population-specific risk variants and the importance of the work in this fine-scale population differences points out, this one on type 2 diabetes really points out that there are just different ethnic-specific differences in the genetic architecture of disease. And I'd like to take this a step further and say, this is a study of African American and European Americans. I would guess that within those, due to some of this work that we're seeing, there are also large differences within the African Americans in these studies. Prostate cancer susceptibility in men of African ancestry. 8q24 is an area on chromosome 8. It's an intergenic region that has been identified time and time again for prostate cancer risk. This paper identified a variant that's only found in men of African ancestry, which is, again, highlighting the ancestry-specific variation. But again, I'd be curious to investigate this and try to see across African American communities what this variant could potentially look like. So this paper really is starting to get at this genetic community structure, and this is looking at the genetics of Mexico, recapitulates Native American substructure, and affects biomedical traits. So here they are really highlighting that accounting for this fine scale ancestry pattern is critical for medical and population genetic studies within Mexico and Mexico descent populations. So to wrap up, from the research from our group, I do believe there are some implications for precision medicine. And I know the large sample size really, we are able to identify some distant ancestry and the more novel recent fine scale population structure. So it is going to take large sample sizes to start to tease this information out. And the population structure can impact findings in different types of genetic research. So this work I do think is relevant from variant identification in a GWAS setting all of the way to clinical trials and response to treatment along those lines. So just again, I wanted to highlight that some of thinking about this in the context of the work in precision medicine, the population structure can lead to, without taking it into account, could lead to some false positives as well as false negatives, but thinking about it can really lead to a more robust study. And I think everyone has kind of touched on it, but you know, if our work can be in groups or considering groups where the results will be more relevant to all, not just the population it was identified in, I think in precision medicine we would have the ability to help a lot more people. So with that, thank you very much for having me.